Hey everybody. So this is going to be a quick video over the other major chemical sense, the gustatory sense, or our sense of taste. So let's get right to it. All right. So we are sensitive to 5 different taste sensations. You see them listed there. They're ones that, at least the first 4 are ones that I'm sure you can easily remember tasting. Um, the 5th one, unami, is kind of that meaty or savory uh, taste that you sometimes feel taste when you're like eating a steak or something like that. Now, obviously, saltiness going to come in there. Certain aspects of bitterness or sweetness are going to come in there. Maybe even some sourness. But it's that meaty, savory taste of what unami is. Now, we traditionally have taught and have thought that we had that different areas of the tongue were sensitive to the were sensitive to different tastes. Now, although you can kind of think of this as being largely still true, it's not as cut and dry. So, for example, if you look over here, here's a rough map. This isn't the this and this are the only parts of your tongue that process salty taste. There are other receptors that process the taste of saltiness spread across the tongue. Now, but the largest concentration of are back here. The largest concentration of sour receptors are here, etc. for the others as well. So it's not that reception of that taste is only localized to these areas mapped out here. They're spread across the entire tongue, but there's a higher density of those receptors at various areas across the tongue. Now, when we talk about these receptors, by and large, oh, sorry, I thought I was going to talk about how the receptors work. Where are they located? So your taste receptors are kind of bundled together into these buds, like this. So each of these are receptors, okay, and they are bundled together into a. So this is one big taste. So this is one taste bud. So you got your taste receptors bundled together into these buds. Now each of the receptors have from one end little hair cells called microvilli. And these villi extend from the taste cells, the taste receptor cells, up into these openings in your tongue called taste pores. So you've got these pores and these little micro hairs, micro hairs sticking up in those pores. Those the microvilli in the pores have the receptors for the various chemicals that you taste. Now, those taste buds are housed in these protuberances on the surface of your tongue. So if when you look at your tongue, you'll probably notice that it's got all these bumps, just tons and tons and tons of bumps. Those bumps are papillae, all right? So embedded on the surface of those papillae, you've got those taste buds, you've got your taste receptor cells, and those taste receptor cells are bundled together in buds, and they have their microvilli with the receptor sites on extended up into the pores in the papillary surface, on the surface of your mouth. All right, and so you have these. That point is a little bit misleading. Sorry about that. The taste buds aren't suspended in the mucus. The microvilli are suspended of the mouth. right? Now let's talk about the receptors. Now I know there's a lot on the screen. I'm going to try and get my photo out of the way as best I can. So remember that we have, sorry, we are sensitive to 5 different taste sensations. Well, we have different receptor processes for some of those. For 3 of the 5, bitterness, sweetness, and umami, they function basically the same way that your olfactory system do, that the olfactory receptors do. In fact, that's not the first similarity. If you notice, coming back, right, talking about those taste receptor cells with microvilli extending out into the into a pore and being suspended in mucus and being there for the chemicals to attach to the receptor site, just like the olfactory cells. And so the receptor sites, several of them work much the same. So, for example, they have the signal proteins embedded in the wall of the microvilli. When the right chemical with the right molecular structure comes along and binds to the receptor site, it releases the G protein, triggering a similar process to what happens in the olfactory system, causing sodium channels to open, letting sodium come in, and triggering an entry. Basically the same process that happens with your olfactory system, with your olfactory receptor. Now, bitterness, we're seeing a lot of chemicals taste bitter to us. And for that, also, so, here's the standard one, but here's, sweet and sugar, right? So here's these 3 listed them. They're listed here, but you see them up here. Now, you'll notice that bitterness is here and here. So this, right here, that's that process I was just talking about. Signal protein embedded in the wall of the microvilli. Chemical comes along, attaches to the right receptor, triggers the release of the G protein, triggering a similar process, ultimately opening sodium channels go out and triggering a potential. But bitterness is also triggered by receptors that are more, you can think of it as being more similar to inotropic receptors in the brain. So instead of having this G protein, so we have some some receptors for chemicals that we perceive as bitter that involve this standard G protein coupled process. But bitterness can also be triggered. We can also taste bitterness by certain chemicals like quinine. When the chemical comes along and attaches the appropriate receptor, it opens up a sodium channel directly and allows sodium in. So you don't have this whole signal protein. You just have a sodium channel that's gated by the chemical. And saltiness and sourness have a similar system to that. So with our perception of saltiness, the receptors for saltiness are triggered directly by salt. All right, so when salt comes in, it goes into the sodium channels, and it triggers your an action potential, allowing you to experience the sensation of salty taste. Sourness works by hydrogen. So you'll have a hydrogen ion come in, activate a channel, and that allows positively charged ions to flow into sour receptors. Again, depolarizing the cell, causing an action. So, for 3 of the 5 sensations, we have that standard G protein coupled process that we have in our olfactory system. Bitterness bitterness is one of those three, but it also can be triggered by activity at more ionotropic type receptors. All right, so you just have, instead of having to, kind of like a ligand-gated channel, like a scissors. But that makes a certain degree of sense because bitterness is typically caused by things, the sensation of bitterness is often caused by stuff that can be, by chemicals that can be harmful. So it makes sense that we would have multiple ways to represent bitterness, which signals to us, hey, that get that thing out of your mouth because it's not good for you, or potentially not good for you. And then saltiness and sourness, they have that more ionotropic type receptor, meaning they have just ligand-gated channels. Saltiness, sodium comes in, blood then takes sourness, hydrogen comes in, triggers the opening of a channel. Positively charged ions come in, depolarize and stuff. And you taste sour. You experience sour. All right. So there's another similarity between the two major chemical senses, how they represent or how they react to the chemicals. Now, what happens once the olfactory sensation, the olfactory information is transduced at the receptors? Well, it then gets carried from the tongue up to the brain, but it gets carried through three different tracts. So the back 2/3 of your tongue, the information coming from those receptors gets routed to your brain through the facial cranial nerves or cranial nerve 7. I say the back 2/3? I'm not 2/3. The back of your tongue gets routed through cranial nerve 9, and then the back of your oral activity gets routed through the vagus nerve or cranial nerve 10. All right. Now, that information all then goes to the medulla, and largely localized to the solitary nucleus, or as I've mentioned here, the nucleus of Tract solaris. Now, that's kind of the gustatory nucleus, or that is the gustatory nucleus of the medulla. From there, it gets routed up the central tegmental tract. Remember, central. Remember what the tegmentum is. You know what a tract is, right? So it gets routed up the central tegmental tract up to the thalamus, specifically the ventral posterior nucleus of the thalamus. Now that may ring a bell because I mentioned it back when we were talking about the thalamus and about the different nuclei of the thalamus. And we'll come back to it when we start talking about the mechanical senses. And that, if you're a nerd like me, that might intrigue you because if you remember, the ventral I just said that we're going to come back to it when we talk about the mechanical senses. And I mentioned back when we talked about it in the neuroanatomy chapter, that the ventral posterior nucleus of the thalamus routed tactile information. Well, that information from the tongue goes through the ventral posterior nucleus, and it goes to several areas, one of which is the somatosensory nucleus, or the somatosensory cortex. So one cool thing, it's cool to me, the palpulae on your tongue they developed from hair cells, all right, and they kept this connection to the tactile sensation of your brain. So I don't know if you're anything like me, but there are a lot of foods that I like just because of the texture. There's very few foods that I dislike because of the texture, but I've heard people who don't like some of them. I guess I don't like oysters because of the texture. But we're experiencing that texture because of the information from the tongue being routed through the ventral posterior nucleus to the somatosensory cortex, where we process the touch or the tactile components of the things we eat. But the primary gustatory nucleus is in the insula. And that's where we process the basic features of the taste. And then it then gets routed, as you can see here, onto the orbital frontal cortex, which you can think of as the secondary gustatory cortex. Now, remember, that's also an area that processes smell. So here's an area of overlap. Now, there's also areas of overlap uh, down in the pathways. So these are reasons why smell and taste can be heavily interwoven. But there's a common misconception that they are the same sensation. They're not. All right. So your sense of taste is not your sense of smell. So if you lose your sense of smell, you can still taste. Your sense of taste will be impaired. Things won't taste as good as they did when you had smell. But they are independent, although interacting sensations. Your sense of taste is not your sense of smell. All right? They are independent. And that, my friends, is that for the gustatory sense. That's it for this video. As always, if you have any questions, let me know. If not,